Well, as you gathered, I'll be talking about how I eventually got to look at viruses and what that all means and where it comes from. I was actually, as an undergraduate in London, I learned physics and mathematics. I had no knowledge of biology or even chemistry, but things have changed. When I got to Glasgow as a student there of Monteith Robertson, this structure was the first structure which I solved with X-ray crystallography. As you can see, there are about 20 or so carbon atoms, and our interest in that structure was mostly what is the distance between bonds, partial bonds, and getting the relationship between bonds and bond order and bond length. But that taught me something about crystallography, and I then actually spent some time in Minnesota learning something about computing. So by the time I got to Cambridge, I had some knowledge of crystallography as well as computing. I got to Cambridge because in, I think it was something like 1957, there was a meeting of the International Union of Crystallography in Montreal, which I attended. And I heard Dorothy Hodgkin speak about the work going on in Cambridge. And that interests me from the purely mathematical point of view, how to solve something as complicated as a protein. And so I wrote to Max Perutz, got accepted. And at that time, for instance, my major professor from Glasgow indicated to me that he thought that what Perutz was doing was not really science, he told me. And there are reasons for that, what Perutz was attempting to do on hemoglobin was really beyond imagination at that time. But by the time I got to Cambridge, John Kendu, who was a student of Perutz, had determined the structure of myoglobin at 6 angstrom resolution, and this is what you see here. And these sausages in this structure were thought to be, and correctly thought to be, alpha helices, as had been predicted by Linus Pauling based on the structure of individual amino acids. And the atmosphere in Cambridge at that time was very stimulating. The whole group of Max Perutz had been turned out into this hut at the foot of the Cavendish, Austin wing of the Cavendish, and it was a very interesting group of people in the, let's see, yeah, in the room behind here. This was the room of Francis Crick and Sidney Brenner. Perutz's office was just next door. Us postdocs were on the other side of the hut. And every morning we had coffee at 11 o'clock, and this was one of the morning coffee times. And the discussions of what things were in biology was highly stimulating, including, as we shall talk in a minute, what virus might be. And because Crick and Watson had determined the structure of DNA, and that gave some idea of how much information there might be in the genome in a virus because of the volume of a virus. But during all this, my task was to help Perutz on the hemoglobin structure, and one of the problems was how to find the heavy atoms. I won't explain this diagram, except you can immediately see that there are nice peaks, and that therefore was very easy to interpret. Is a function which I invented, and was very clear how the heavy atoms could be determined in the isomorphic replacement, and that was an essential part for determining the phases for the structure determination of hemoglobin, which is shown here. This was one of the sections through hemoglobin, and there again, immediately you see the sausages, and Max had the good idea of making models in this way. For instance, this is then the model of hemoglobin, and you can see the long alpha helices. Each layer, each section, like I showed on the previous slide, is shown here as a actually piece of clay molded into the right shape. And taking the part, we see the 2 alpha chains and the 2 beta chains, each related by a 2-fold axis. And then the stunning thing about the whole discovery of this was that myoglobin, which I just mentioned earlier, had been done at 6 angstrom resolution, and then actually that same year at 2 angstrom resolution, and the alpha and the beta chain of hemoglobin all had the same fold. This was the first time that evolution could be seen at the molecular level, where things really happen and develop in biology. And that discovery has guided me for the rest of my life, as you will see. Now, in addition, seeing the relationship between the alpha and the beta chain in hemoglobin made me wonder whether that could have been discovered without all the work on heavy atoms. And as you see here, I wrote in really the first paper on what's now called molecular replacement. I said evidence is accumulating that many of the larger protein molecules are made up of identical or closely similar subunits. The reasons for expecting this for the protein part of virus structure were set out by Crick and Watson. I'll explain that more in a minute. And the prediction has been amply confirmed in crystallographic studies of spherical viruses, as is shown here. Oh, sorry. Here, this is work of Don Casper in Cambridge, where he shows the angles between the symmetry axis being represented by strong intensities on the diffraction pattern, correspond to that of what would be in icosahedron. But what Watson and Crick had suggested, as I guess you probably all know, that because of the limit of information in a typical genome of a virus, it must mean that the capsid of the genome must be repeated many times to cover the genome. And therefore, you have the problem of each identical subunit would have to have, therefore, identical environment. And that can only be had if you go into a regular polyhedron, like a tetrahedron here, and octahedron, icosahedron. Icosahedron being the most complex of the regular polyhedra, and therefore they were suggested correctly that this might be the symmetry of a typical virus. And as you saw, this was also shown in the case of a plant virus, tomato bushy stunt virus. In those days, plant viruses were the one things which could be studied better than other viruses, because it was easy to grow the viruses on plants in greenhouses. Now, that stimulated my mind, as I said, in Cambridge was a place to think. And so I really wanted to use this idea of molecular replacement, which I've just partially explained, to look at viruses. But the idea of studying viruses in those days was beyond what would likely be funded. So I decided to work on proteins or enzymes which would have some kind of symmetry, similar to what would be in a virus. And that led to this structure, nucleotide binding structure, which in fact Neil mentioned in the introduction, 6 parallel beta strands, and the nucleotide would bind at the carboxy end of the sheets. And that structure, which we discovered then on looking at various dehydrogenases in the late 60s, early 70s, is probably the most common fold found in all structural and now also in genome sequence, because you can recognize it by the sequence of amino acids or sequences of nucleotides. With that discovery, then I felt it was time we could start working on viruses. And as I said, we started working on plant viruses. And the first plant virus structure was determined in the early 70s. Steve Harrison in Harvard had also been doing similar things. And so we had more or less at the same time determined the structure of a southern bean mosaic virus in Purdue and tomato bushy stunt virus in Harvard. And these two structures, these two different plant viruses, RNA plant viruses, turned out to have very similar structures. Now, I should have mentioned that although an icosahedron only has 60 subunits, it was soon recognized that there would need to be more than 60 subunits on the surface of the virus. And Casper and Klug suggested schemes in which identical subunits could be arranged at least in quasi-identical ways. And so if there were, say, not 60, but 3 times 60 subunits, that would be triangulation number 3. And these plant wires had a triangulation number 3. You can see in the triangle, this is the icosahedral asymmetric unit, and we have 3 different yellow subunits with this fold. They were covalently identical sequences, but they were arranged in this way. And then another 6 or 7 years later, in about 1985, we were able to determine the first plant vase structure, which was a common cold vase. And this is shown down the next picture down here. And in this case, the 3 quasi-identical subunits here, which had the same sequences, were quite different sequences, but nevertheless had more or less the same fold. So again, we have, as we found in hemoglobin, similar folds are conserved over much longer periods of time than either sequence, and gives you some measure of evolution over long distances of time. And there were other things which are very interesting about the structure of the common cold vase or Reiner vase, which we determined. For instance, it had a deep cleft, or we call it canyon, around each 5-fold axis. And then here we have the 3 different subunits, viral protein 1 in blue, 2 in green, and 3 in red. Working together with Roland Ruckert at the University of Wisconsin, Barbara Sherry, his student, they found that the site at which they found the amino acid at which certain neutralizing antibodies bound. And then when we had the structure, we found, as we might have expected, that these sites were on the outside of the vase, and these blobs here represent the binding site of these neutralizing antibodies. And now we have the canyon, and that suggested that the depth of the canyon, which is shown here, might be the site of receptor binding, because these residues were not changing. The places where the virus was changing most was where it was binding the neutralizing antibodies, and this is the antigen binding fragment of the antibody. And so this was a prediction which can be called the canyon hypothesis, which might be applicable in other cases as well. And we then were able to confirm this using, for the first time, electron microscopy. I had gone to Cambridge in 1980 and discovered, well, not discovered, learned something about electron microscopy. When I came back, we were ready to look at these viruses. And the nature of the receptor for the Reiner viruses was determined by Tim Springer, for instance, in Harvard, and was the intercellular adhesion molecule 1. So we were able to obtain some ICAM1, intercellular adhesion molecule 1, bind it to the virus and do an EM, electron microscopy, very low resolution in terms of today's ideas study, but we were able to show that the canyon hypothesis was indeed correct, and we were able to see where the orange ICAM1 bound into the canyon. There are other interesting things about the Reiner virus structure. What we see here is actually viral protein 1, and we came to call these folds jelly roll folds. I'm not sure why. I think it was Jane Richardson who worked out this name. But if you name the strands, beta strands along the polypeptide chain A, B, C, D, and so on, then the jelly roll fold consists of 2 anti-parallel beta sheets, and one of these would be BIJ and the other one would be CHEF. So anything like this would be a conserved jelly roll fold. As we'll see, this fold is very frequently used in the structure of viral capsids. And we also found that working with people at Sterling Winthrop Company, that there were small molecule inhibitors, and these inhibitors bound into the hydrophobic area of the central of this beta bell fold. Now, the center of this must be hydrophobic in order to fold up as to exclude the water. And here is one of these compounds, Win51711, which we found bound in this. And they stabilize the virus, and that's how they work. The virus could not uncoat and also could not attach easily. We continue to develop, together with a subsequent company called Biopharma, antiviral compounds, developing a compound called pleconazole, but in the last stages of its development, the FDA did not license it because it had unacceptable side effects on women on birth control hormones. Nevertheless, it can still be used in compassionate cases, and this quite long ago, the number of people who had been brought back from almost death to life in a few days by the use of pleconazole was quite significant. Now, rhinovirus, which is a picornavirus, small RNA virus. There are other more complicated viruses. So we went on to look at, together with Richard Kuhn, who joined us at Purdue, look at more complicated viruses such as flavivirus and togaviruses, both of which use a membrane to envelop their genome, and then there's a capsid protein outside that. And typical of flavivirus, I think, is probably all of you will know, is dengue virus, West Nile virus, yellow fever, and Zika virus. And togavirus, typical, is Symbis and Venezuelan encephalitis virus. And more recently, chikungunya virus has been very much in the news. So these viruses undergo, the flaviviruses undergo, like all viruses, a life cycle. There is an immature particle, which is shown on the left here, and a mature particle. And the difference here is very spiky, as you may see, and these are electron micrographs, and this very smooth mature virus. So clearly something is happening to the virus as it goes from immature to mature. Now, most viruses require a maturation step, because in the cell in which they're being assembled and being made ready to leave the cell, they obviously must not infect their own cell. And so usually there is a proteolytic event as the virus leaves the cell to arm the virus to be infectious just as it's leaving the cell. But this mustn't happen before, and that's the maturation process. So within the cell, say dengue virus is quite different to what happens as soon as it's outside the cell. We then were able to, in those days, this was about 2002, we were able to get a 26 angstrom resolution map using electron microscopy. The big problem really wasn't the electron microscopy, but getting pure enough virus to be able to do the reconstruction. And you can see here on the right, you can see the membrane double layer. The black is high density, and then there's a glycoprotein outside and the core protein inside. And to do this, we assumed icosahedral symmetry. In the icosahedron, as you've seen, there's an asymmetric unit, which is shown in outline here. There's actually also a twofold axis at that point. And if you know that what the structure is within that icosahedral asymmetric unit, by virtue of the icosahedral symmetry axis, you then know the rest of the virus. Taking a cut through the virus, or diagrammatically, you can see the double layer here in green, lipid bilayer, and the core inside, and the glycoprotein on the outside. So this is what a flavivirus turned out to look like. And at that time, Felix Ray, working in Steve Harrison's lab, was able to crystallize the glycoproteins. Now, Franz Heinz in Vienna had shown that with trypsin treatment of tick-borne encephalitis virus, which is not a flavivirus, you could remove the glycoprotein, and this is what Felix Ray did, and was then able to crystallize it. And he found that here we have it's a dimer, and one monomer consists of domain 1 in red, domain 2 in yellow, and domain 3 in blue. And you can also see a green part, which is about 12 very hydrophobic amino acids. And these are called the fusion peptide, because these would be known to be used to the hydrophobic part to enter the cells of a possible host cell. So as I said, we were able to determine the electron density of dengue virus, in fact. And then there's a question of how to interpret this. You can see here was the fit of one dimer of the dimer which Felix Ray had determined of tick-borne encephalitis, well, different virus, but we assume that possibly there's homology between these different viruses. And what I did is to show this works. The dimer has a two-fold axis. I placed that on the icosahedral two-fold axis and then returned it around here in all possible directions, slipped it up and down a two-fold axis until it fitted. I did this by computer and showed that this was the best fit. Now, you can see some part of the density is still uninterpreted. You can probably guess what this is, and here it is fitted. So now we have the total structure. We have the icosahedral asymmetric unit. So that gives us a total structure, which we can see here in the same color code as I explained originally. And that means that there are 3 monomers per asymmetric unit, which you can see here, green, blue, green, red, and blue. The green and the red one are is the dimer in the general position, and the blue one is the dimer in the special position, which I explained to you how I fitted in the first place. Now, the problem with this structure was that the monomers, each monomer has a different environment. This goes against the ideas of Casper and Klug, which said that each monomer would have at least quasi-equivalent arrangement. So this was one of the 2 reasons why the referees in Nature rejected our paper. We actually did manage to publish this in Cell, but I like to tell this story because it has happened so often in my life that the referees don't like what I do, and we have to publish somewhere else, which I find rather annoying. Now, going back to the structure, and this is the electron density at the level of the membrane, and the black part is again high density, and this is where the helices, transmembrane helices in it. We are able to interpret that. We take that part and enlarge it. And now we see that it's superimposed here. This is a triangle, again, icosahedral asymmetric unit. This is in pink is the dimer in the special position on the two-fold axis, and in blue and in green is the dimer in the general position. And you can see, take, for instance, the pink monomer, that 4 transmembrane helices to that monomer, and in green, by translation, you see exactly the same transmembrane helices for this monomer. So that made it a very reasonable interpretation. And the blue is again the same, except there's a molecular two-fold axis at this point, icosahedral two-fold axis at that point. So diagrammatically, we have now the structure of the mature virus. We can see the 4 transmembrane helices per monomer, and this is the 3-domain structure of that monomer, and here is the dimer more diagrammatically. As I said, there's also an immature virus, and the immature virus had spikes, as we have already seen, and these spikes could be interpreted in quite a different way. Here's one of those spikes, and this is the structure of the monomer fitted into each spike. And now the fusion loops, and remember this is the immature virus, the fusion loops are 3 of them here on top. So this is a trimer with fusion loops here, but there's uninterpreted density. And this is the immature virus, and in order to become mature, there's a proteolytic event which removes the pre-peptide from what's called the pre-M membrane protein. So this is, at the top here must be presumably the pre-peptide, and that was exactly what it turned out to be, which protects the fusion loop from being available while in the immature virus. And when this is removed, the pre-peptide at the top here is removed, the virus is then infectious. Ju Chen at Purdue was wondering whether we could do this maturation process in vitro. So she used furin in vitro, which is the cellular enzyme, and she showed that at high or neutral pH, she was unable to cleave the PRM. But at acidic pH, with the furin, she could get the lower molecular weight, PR and M protein more or less on the same position on this gel running apart. So she was able to do the cleavage at low pH. And that was also consistent with the structure because the site of cleavage was within the trimer and unavailable, therefore, to the furin, sterically hindered from cutting it at neutral pH. So what happens here, as she interpreted, here we have the virus being assembled in the endoplasmic reticulum, and then it passes through the Golgi and finally through the trans-Golgi network. And that's where the virus encounters the acidic pH in the cell, and therefore that's where the furin can be cleaved. So here we have the trimeric structure in the ER. The red part is the fusion peptide on the E protein, and then as it goes through the Golgi, as she in fact Ju had shown, the virus changes the trimers. The 60 trimers are arranged into 90 dimers, and that cleavage can occur. But the PR here doesn't come off. It's kept there because the pH is low, and the interaction in the PR, and as we had shown structurally, and the E protein is a series of ion salt bridges. Eventually comes off, and the virus emerges and goes back into the neutral pH of the extracellular environment, and you can somehow this pointer is decided to give up at the ghost. As you see on the top diagram there, the PR comes off. I don't know what's happening here. I think I have to give up on the pointer. This is shown diagrammatically here, where we get the timer spike, and then this is reversible, amazingly reversible. But when the pH is low, the furin can cleave, and you watch the pH meter, the PR comes off. Oh, wonderful. Thank you. So now we got a red pointer. Well, I don't know. Maybe I'm doing something wrong with my hands because I can't see it. Never mind. Let's go on. So we've seen how these flaviviruses mature. But there are, of course, 60 places on the virus which has to have maturation. And what we're requiring is that furin comes along in 60 different places on the virus and cleaves all those places to make the virus mature. So Pavel Plevka in my lab, he was a postdoc, is now in the Czech Republic with set up his own lab. You can see in the center there a cryo-EM picture of a typical mature virus, which is still a little bit funny on one side. And he looked at this with tomograms, which in tomograms you don't take the average of thousands of particles. You're looking at a single particle, and the colors here show the degree of correlation between the mature structure. Take the left-hand picture. Between the mature structure, the red is good correlation, the blue is bad correlation. And if we, on the right picture, we can see the correlation with the mature structure. And so what we see on this particular single virus, that there is actually, the virus is more immature on one side and more mature on the other. And that actually was a major problem when we came to look at Zika, because these viruses are then, a mature virus is never completely mature in these flaviviruses. And when you do electron microscopy, you're dependent on having homogeneity, that you can average the different particles. And that was a big problem when we came to look at Zika virus, which is also a flavivirus. In Zika virus, this was just in the beginning of 2016, beginning of last year, we did this. And in a period of just 2 months, as opposed to a few years for all these other things, using electron microscopy, we were able to show that Zika virus has a very similar structure to, say, dengue virus and the other flavivirus, West Nile virus. Now, I mentioned the problem with the maturation problem. This was a much bigger problem with Zika virus. So we had to use cells which have overexpressed furin to make sure there's enough furin to get more or less mature. And then using the new technique in electron microscopy, we use direct electron counters. We were able, as we can now do quite generally with electron microscopy, obtain a structure at 3.8 angstrom resolution, which you see here for Zika virus. And this showed for the first time a direct determination of a flavivirus, as opposed to the mixture of electron microscopy and crystallography, which we had used in all the previous studies on flaviviruses. Now, Zika virus was also interesting in that it was similar to dengue virus, and what's shown here is the amount of similarity. Well, I can't know what's happening on here. No luck. So we have now to find the similarity of Zika virus and dengue virus. There are 3 independent monomers. So monomers A, B, and C. So if we take on the top line is Zika virus monomer A compared with the 3 monomers of dengue virus, each in a different color. And you can see in the middle where it says 145 to 165. That's where you're plotting the difference in angstrom between the equivalency alpha atoms. So you can see that's the major difference there between Zika and dengue. There are other differences on the right-hand side, but we won't talk about those just now. And this happens in each of A, B, and C, the 3 different monomers of Zika virus. So what is this difference? It's actually at an asparagine, 145, which is glycosylated, a sequence which is enzymatically recognized and is then glycosylated. And here, if you go to the map of Zika virus on the left, hang on. Zika virus is, as I said, we did electron microscopy, and on the left there is the graph of the correlation between 2 data sets, 2 equal data sets, and the virus was, this is a standard procedure, determined from each data set, and you can see the correlation as a function of the resolution at the bottom, and you can see that the resolution is 3.8 angstrom. What you see in the middle of the diagram is the fit of the structure in the area, fit of the structure to given amino acids, and fits pretty well. You can see a tyrosine 203 fits, the arginine 139 fits. Working?
Yes.
Wonderful. Yes, working. Thank you very much. And a very nice tyrosine there. So this was nice density, which we got at near atomic resolution for Zika virus, which we had never achieved before for other flaviviruses. And what we see here is also sorry, I have to go back a minute. I was going to tell you about the differences. The difference corresponds to asparagine 154, which glycosylated. We knew from work which we've done on dengue virus, which uses DC sign as a receptor, molecular receptor, that asparagine 154, which is glycosylated because of the sequence there, that this glycan is involved in receptor binding. And this is exactly where Zika virus differs from dengue virus. When you expect some differences in the virus, what kind of molecular receptor it goes to, because there are different pathological effects. So here then it's clearly, it has a different way of recognizing receptor, different kinds of receptor involved in Zika virus. So this is a very fundamental basis difference between Zika and dengue. We also now looked at structure of Zika virus bound with antibodies, and this is one antibody, Zika virus 117, and we found the red part. This is colored radially. Red is a very large radius, different places where you get the Fab, the antigen binding fragment of an antibody was bound. This is near the two-fold axis, that's why we called it 2F, and that's near the three-fold axis, why we call it 3F. Now, we'd expect 3 such sites, because there are 3 independent monomers, and each monomer would have the same binding site. So we would expect 3 Fab binding sites. But the third site, which is actually nearer the 5-fold axis, was not very big. And we could easily see that this site was overhung by surrounding structure, and they were sterically hindered. Fab was sterically hindered from binding there. We also found that the Fab binding here and here, when you look at it, the footprint, was also overlapping. This is the icosahedral asymmetric unit here. That's the two-fold axis, icosahedral two-fold axis. And the blue is one monomer, the red is another monomer, green is another monomer. And you can see this is one binding site, the 2F site near the two-fold axis. And this is the 3F site near the three-fold axis. And these are physically overlapping. So you cannot bind this antibody to all the sites on the virus at the same time. For instance, if something is binding at the 3F site, then you can't bind at the 2F site because they're overlapping. But at the 2F prime site, which is the icosahedrally two-fold related one, you can bind and not there. And these are all possibilities, which means on average, this site near the three-fold axis is occupied 2/3 of the time, and this side is occupied 1/3 of the time. And experimentally, we found the height of the density of the Fab was corresponded to those things pretty well. So now we understand why the antibody can only bind at certain sites and can only bind basically on average 1 antibody per icosahedral subunit. And yet it completely neutralized the virus, because what the antibody was doing, you go back a minute, was cross-linking. The antibody sites were overlapping from one monomer to another, where they're cross-linking the monomers and therefore binding the monomers so it couldn't fall apart. So we understood then why the virus was neutralized by the virus, because the antibody stopped the disintegration of the virus. And we also understood why it was a very potent antibody, because you only needed a small number, 1 out of 3 binding sites at any one time for the virus to be neutralized. Sorry, going a long way. Now, there also, as far as we have time, we don't have much time left. We looked at different kinds of viruses. We looked at very large viruses, have been looking at very large viruses like PBCV1 and pox virus in general, Mimi virus, very large virus. This shows PBCV1 structure. Again, there isn't too much time. But what's interesting here is that the structure of these very large viruses is based on the jelly roll fold. But 2 jelly rolls within one polypeptide chain are joined together to make this hexamer. This hexamer corresponds to the kind of hexamers predicted by Casper and Klug. And you find this, for instance, in adenovirus. You find it in PBCV. You find it in a phage PRD1. You find it in a plant virus. So this fold is very generally used, but not always used. For instance, in flavivirus, it's not used in the capsid structures. And PBCV1 is also interesting because it's not completely icosahedral. There's a special vertex spike, which is used for entering the host, penetrating the cell wall of the host. And now I want to say just a few things about bacteriophages. We've done a lot of work on bacteriophages, in particular, for instance, on T4, 529. 5X174 is interesting because it doesn't have a tail, or so it appears, but it actually makes a tail during infection. And we have studied these phages, bacterial viruses, by looking at the individual proteins, and then, as we did, for instance, for flaviviruses, fitting the proteins into the lower resolution electron microscopy. And here we have T4. I think probably most of you will have seen similar pictures of T4. This is the head where the genome is kept, a contractile sheath, a tail with a sheath, contractile sheath around it, and the long tail fibers which recognize the host. And now this is a movie made of knowing what the structure is, bubbles in the water, and that's the phage coming down and landing on top of the E. coli host. The long tail fibers are going to recognize the host here. This is the shadow of the virus on the top of E. coli, and then when everything's attached, the base plate unravels and the short tail fibers attach firmly, and then the tube is pushed through with a sort of drill motion through the cell wall. There's also lysozyme digestion of the cell wall. These are the lysozymes falling off there, and then the tube comes through and pushes the DNA in there. And so I want to thank my mentor, and who really educated me about science. He showed me that science is interesting for what we want to find out, not just because it's a mathematical challenge. He was very keen on pointing that out. And then all the people in my group about half a year ago, and I don't really have time to introduce them, but just like I showed you about Cambridge, the point about having a group of people working together at any one time, that they help each other and talk about the problems, and therefore recognize the problems by their discussions. There, of course, have been many people involved in all the work which I've shown you. These are for picornavirus, for the flavivirus, particularly for the Zika virus, and for large DNA viruses, and for the bacteriophages. And thank you very much.